I would like to thank the organizing committee for this event and this opportunity for me. So let's start. I'll tell you a story about polyribosomes and ribosomes. I am from Institute of Protein Research of Russian Academy of Sciences. The polysomes is a large assemblies of ribosomes that translate genetic code and synthesis the proteins. But what do you know about polysomes? They can form many shapes like these. They may be helixes or circles or zigzags and many other forms. And nowadays we don't know how they work. So in order to ask on this question, we had to find the method that allows us obtain structural and functional data at once, both. So we did. It was super resolution microscopy. We armed by this and starting our business with ribosomes and polysomes. What is a super resolution microscopy? There are several types of super resolution microscopy, but we used storm or palm microscopy and how it works. You can image your sample by wide field microscope. So in this case, you excitate all the fluorescent molecules and have this blurred fluorescence. In the case of super resolution microscopy, you can exceed 1 fluorophore per time. So either way, to obtain all positions of our molecules of interest, there is organic dyes like these, cyanine dyes or another dyes. They can be stochastically switched from fluorescent form to dark form and back. So this is a basis of storm and palm microscopy. To obtain the fluorescent super-resolved pictures of ribosomes and polysomes, we had to label the ribosomes. This is a simplified protocol of labeling. We add in organic dye like Alexa or anything else to the ribosomes, incubating. After, we need to bind 3 dyes, 3 molecules of dye, and then we have labeled ribosomes, purified labeled ribosomes, and we can visualize them. But our first tries to visualize ribosomes were inconvenient. All fluorescent signals were blurred, and we can explain this. So we must bind ribosomes or polysomes to the surface to avoid the thermal drift or other cause of blurring the fluorescent signal. this is the best way we have found. The molecules that cause 3-aminopropyl trioxysilane can bind the amino groups of ribosomal proteins. So this is a way to fix them. We choose the model objects. This is E. coli ribosomes. This is a simple object to visualize in. So you can see each ribosome is good, and further we visualized the ribosomes in dual color mode and store microscopy. In this case, you can see even subunits apparently. Green indicates small, 38 subunits, and red indicates large, 50 subunits. It works, you see. But our goals is to visualize the polysomes, to study their functions and their life. So we need a polysome. How we get it? We used Ribosomes extract to obtain polysomes from labeled ribosomes and supernatant fraction. This is a fraction of light proteins, translational factors, and other to reconstitute Ribosomes extract. This is the extract like an initial extract, but with modified ribosomes. So we translate this, and we can see that activity of the reconstructed extracts, red curve, is the same as activity of the initial extracts, blue curve. As for polysomes in these extracts, the picture the same. This is polysome profiling. These peaks indicates full eukaryotic ribosome. This is a large subunit peak and small subunit peak. And this is our polysomes. So we see the loading of polysomes, the same in both cases. It was great, and after that we can visualize them by the electron microscopy. It's micrographs of polysomes negatively stained by uranyl acetate. So we can study the polysomes by cryo-electron tomography or by fluorescent super resolution microscopy. And we visualize the polysomes from Ribosomes extracts. They are. And for comparison, I put on this slide these models obtained by cryo-electron tomography. So you can observe same types and shapes of polysomes. But there is a disadvantage. Serial localization signal is bright and sharp, and other signals are blurred. So what is the reason of this? We used organic dyes to label ribosomes. And character of this procedure is stochastic. Each ribosome can contain and expose on the surface different amounts of fluorescent molecules. So we thought, would we change the labeling strategy. Organic dyes are good, but they have disadvantages like non-specificity, needing a long time to collect required signals for super resolution localizations. And there is another strategy to visualize proteins and of course ribosomes and polysomes. This is genetically encoded proteins or tags. The advantage of this approach is clear. We have only one or two is controllable signals per ribosome, and the amount of fluorescence from each molecule is the same, identical. So we have tried this. We used HIC-293 human cell line. It was transfected by this plasmid that encodes photoactivatable red fluorescent proteins fused with alpha-tubulin. So transfected cells were lysed, and translation in polysomes was stopped by adding the cyclohexamide. That antibiotic stop translation by incorporating in the E site of ribosome. You can see this picture. This is our protein of interest. It can emit fluorescence. Yeah. The nascent peptide chain is stopped. So we visualized these polysomes. You see, all signals are the same, intensity are the same. So we conclude that this way is preferable to visualize polysomes. With super resolution microscopy, we can discern each ribosome in polysome is good. It was our initial goal. It's our team, polysome team, from Institute of Protein Research. This is my chief, Vladimir Sorokin, and this is Dr. Joanna Fonina. She provided me by structures, cryo-T structures of polysomes. And this is a team of MSU, Moscow State University, electron microscopy facility. We have polysomes, and they have microscope. So we did it. Thank you.
Thank you for the nice talk, Ivan. Do we have questions?
Thanks for the talk. So what kind of information do you plan to extract with palms that you cannot get with cryotomography?
On small, please, echo.
Sorry?
We have echo. On small, please.
So what kind of information can you get with a super resolution microscopy here that you cannot as well get with cryo-electron tomography?
Okay, we can get information about structure, atomic structure, by cryo-EM, but with a super resolution microscopy, we can get information about origination of each ribosomes. From where did she come?
How?
By labeling with different dyes, green or red. Or
Cool. Thanks.
So I have a question in the beginning of your slides. So you said that you use slides that are silanized, right? They are coated by a silane.
Which slide?
The very first one with the cover of your cover slip of your slide. The question is, how does the ribosome binds to the cover slip?
Ah, okay.
Yeah, this one.
So this is a molecule of APTES. After that, we need to activate the surface of cover slip by dialdehyde.
So practically the ribosome binds by any protein on the surface.
So the aldehyde groups are exposed and they bind the amino groups of ribosome proteins.
Okay, so that brings me to my second question. How do you actually know that these complexes are polysomes and not just aggregates?
It's a good question.
I mean, it would be nicer if you
We
had...
controlled that by sucrose gradients, but it's only information about loading of polysomes. And addition control is a electron microscopy. We can see resin aggregate.
Okay, thank you.
Maybe in addition to this question, I mean, you have now some electron microscopy images over there, but are you also sure, maybe I cannot just see it, but are you sure that there is also RNA, or how do you ensure that there is also that they bind to RNA, because in your fluorescence image you just see some spots in the end. So.
We labeled mRNA too, but our microscope has broken. So we cannot see this today.
Okay, and another question, you used HEK cells for your one experiment. Have you also tried other cell lines, or just the cell line, the HEK cell line?
It was the cell line which we have, so.
Okay, are you planning also to do it on other cell lines, or?
Excuse me?
Are you also planning to use other cell lines or also other organisms to find out, maybe? Or just stick there for model at the moment?
In future, we'll try, but not now.
All right, one question over there.
So I might have missed that, but it's a nice method to visualize the polysomes using these recombinant proteins. But what kind of analysis do you want to do afterward? Like, you want to see the dynamics of this formation of the polysomes, or what do you want to see afterward? Now that you have been able to visualize them, what do you want to see afterward?
We have to see each ribosome in polysome, and the original aim is a secret now. So I can't answer for it.
Okay, I think that's it for the questions. Maybe we have more questions. We can find Ivan outside. Now, thank you, Ivan.
Okay, thank you.
Now it's my pleasure to introduce our next speaker, Anne-Claude Gaver. She has been the director of the cellular and molecular biology unit at a biotech company. And since 2005, she moved to EMBL Heidelberg, where she has been at a group